This is our 11th year of offering the Longwood Seminars, and this is our third seminar of 2011. We're delighted to offer you tonight the topic, From Vision to Touch, Exploring the Five Senses. I'm Gina Vild, Associate Dean for Communications and External Relations at Harvard Medical School and Chief Communications Officer. May I see a show of hands for anyone who has attended one of the first two seminars this year? Oh, wow, fabulous, great attendance. Can I also ask for a show of hands if you've attended in past years? Wow, we have many, many loyal fans, so thank you for being here and for enjoying these. I hope all of you are members of our Facebook page. As you may know, this year we offered 11 or 12 different topics and asked our friends on Facebook to vote for the topics in which you had the greatest interest. And this was one of the topics. So I hope you enjoy it, and I hope you will continue to post and stay friends with us on Facebook. If you have ideas for next year's programs, also please offer your suggestions on Facebook. We really do want to hear from you. If there's a topic of particular interest or something we've offered in the past that you think we should offer again. Tonight's topic is really special to us because last year Harvard Medical School launched our Harvard Medicine, a re-envisioning of the Harvard Medical School Alumni Bulletin. The topic for the first issue was the five senses. And we're delighted to have this Longwood Seminars tie in with the theme of that magazine. If you would like to receive an e-copy of our magazine, which is produced 3 times a year, please either post your address on our Facebook page, see a member of our team in the lobby, and we'd be delighted to add you to that list. There's one more seminar left for this year. April 12th, Food for Thought, Genetically Modified Nourishment. We are nearly full, but if the topic interests you, there's still room for a few more participants. So now just a few reminders. We are videotaping this. If you are seated in one of the first couple rows, you may wind up on the video, and it's broadcast throughout the world. If you're not comfortable with that, I ask you to move to the back of the auditorium. If you attend 3 or more seminars, you can receive a certificate of completion. Certificates will be available after the April 12th seminar. You can pick yours up then, or if you're not able to attend, we will mail your copy to you. Teachers, a reminder, you can earn professional development credits by attending all 4 seminars. You would need to complete an evaluation form after each one. Evaluations are available in the lobby. You will see members of our staff with index cards. If you have a question for a member of our panel, please write your question on one of the index cards, and we'll try to get to as many questions as we can. I also want you to know that the videos from the first 2 Longwood seminars are now available online. So if you would like to view it again or share it with a family or friend, we encourage you to go to the Harvard Medical School website. Finally, please turn off your cell phones. Tonight's seminar is called From Vision to Touch, Exploring the Five Senses. Your senses make it possible for you to explore your surroundings. It's how we communicate in the world. We rely on our senses all the time, even when we're not thinking about it, without even knowing how they work. So after tonight, you're going to know a great deal more about how your senses work, because we are very, very fortunate to have 5 extraordinary members of the Harvard Medical School faculty here with us tonight. They will share with us their wisdom and their research on each of the senses. Our first presenter, I'd like to introduce Dr. Bob Data, who will discuss our sense of smell. Dr. Data is an assistant professor of neurobiology at Harvard Medical School. Dr. Data's research focuses on how the brain is wired to extract information from the environment and convert that information into action. Thank you. Welcome, Dr. Data.
Hello. You can hear me? Great. So thanks, Gina. Again, my name is Bob Data. I'm an assistant professor in the Department of Neurobiology here at the Harvard Medical School. My laboratory functions on trying to unravel the neural basis for olfaction, for our sense of smell. So to begin, to the brain, all reality is virtual reality. All animals have evolved senses to extract information from the environment and transmit this information to the brain. But these mechanisms don't extract all or even a small fraction of the information available in the environment. Instead, evolution has selected for specific neural machines that enable us to sense just that which is most critical for our survival and to ignore the rest. We, of course, have our 5 canonical senses, but other species sense that which we cannot. The echolocation systems in bats, for example, or the thermosensory systems in pit vipers enable these organisms to sense high-frequency sounds or heat and hunt prey in their particular ecological niches. And so our perceptions of the outside world, which are deeply shaped by that which we're actually sensing, don't reflect reality. They're constructs, adaptive fictions, selected for by evolution, built by genes, and instantiated by the neurons that make up our brain. So how does the brain usefully organize information in order to enable perception and to promote survival? Well, the brain contains within it internal representations of the outside world. In other words, the brain contains maps that tell it what's out there and give it hints as to how best to behave to solve its particular problems. So let's begin by considering what a map actually is. So this should be a familiar image to you. It's a map that effectively compresses information that's spread out over a very large spatial scale into a kind of a more convenient package. This feat is called spatial transformation. In this case, it takes an object that's 3,600 miles across approximately and compresses it to the size of this screen. The important point about this is that the relative positions of all of the objects depicted by this image, the rivers, the forests, the mountains, are maintained despite the radical spatial transformation that's been imposed here. And so this map is topographically organized. And because of that, we can use this kind of map to usefully navigate this environment. So through spatial transformation and topographical organization, you can effectively organize maps for all sorts of information, for simple physical parameters, like elevation, as shown here, or for much higher order and abstract ideas, like political boundaries, as shown in this grade school map of the lower 48 states. Now, when we as humans look at an image like this, we kind of intuitively and immediately try to interpret the meaning of the data embedded in this map, trying to look at the boundaries and think about what they mean. And artists like Jasper Johns take advantage of this intuition, and by tweaking our expectations, can create a highly effective aesthetic tension, as shown here in this Jasper Johns painting called Map. Now, even though it's highly degraded, we all recognize this painting as a map, that is, as a topographic representation of information about the outside world. And as I mentioned, the brain contains within it many such maps, except, of course, within the brain, information is not splayed across a canvas. It's organized in populations of neurons that are arrayed as sheets within the cortices of our brain. And information in the brain is not encoded through color. It's encoded through the universal currency of information transfer in the nervous system, through electrical activity. So for instance, when we're touched, where we are being touched is topographically organized within the part of our brain responsible for responding first to touch, the primary somatosensory cortex, with the neurons that respond when we're touched on our wrist residing next to those neurons that respond when we're touched on our arm, and so on. And this topographic organization provides an obvious mechanism through which the brain can understand at any given time where we are being touched. Similarly, when we hear sounds, information about the frequencies that make up those sounds are topographically organized in the part of our brain responsible for first responding to and processing sounds, the primary auditory cortex, with neurons that respond to low-frequency sounds being adjacent to but distinct from those neurons that respond to higher frequency sounds. And so our brain contains within it a tonotopic map that topographically organizes information about frequency that helps the brain know what it is at any given moment that we're hearing. And finally, when we gaze out upon the world, objects from the world reflect light that falls onto our retinas in a topographically arrayed manner due to the structure of the eye. And information, this topographic information is faithfully propagated into our primary visual cortex, the part of our brain responsible for processing vision, in a topographically organized manner. So this part of our brain contains a map of visual space that helps our brain know where in visual space any given object is. Now, as I mentioned before, my laboratory focuses on trying to understand our sense of smell, and in particular, it wishes to address the following question. how does the brain make maps for smell? It turns out this is a very complicated problem. If you consider the three senses I've spoken about thus far, touch, hearing, and vision, each of these has an obvious physical axis the brain can use to organize sensory information, like where we are being touched, or what frequencies we're hearing, or where in space particular objects reside. Let's consider a common scent, the scent of a jasmine flower. It is made up not of one odorant, but actually of tens to hundreds of individual monomolecular chemical odorants that are volatile, each of which has a different shape, size, mass, chemical composition, functional group, resonance frequency. And so there's no single axis the brain can use to represent all this very complex information, all of which collaborates to generate the ineluctable scent of a jasmine flower. So how does the brain solve this problem? So I'll tell you what we know. We, of course, smell sense through our noses, which house within it a special structure called the olfactory epithelium. Within the olfactory epithelium, there are specialized neurons called olfactory sensory neurons whose job it is to sense odors from the environment and then trigger electrical impulses that go to the brain. So the way they go to the brain is they travel through nerves called axons that terminate in a structure at the base of the brain called the olfactory bulb within specialized balls of neuropil called glomeruli. And I'm going to show you some images of glomeruli in a few slides. Information is then sent forward from glomeruli to multiple regions in the primary olfactory cortex in parallel, and these multiple regions collaborate to generate the coherent unitary percepts that we associate with smells, like the aroma of coffee or the scent of this flower. If we look in a little bit more detail, we learn that individual olfactory sensory neurons express special proteins called odorant receptors, which are molecules that sit on the outside of these cells and actually physically interact with odorants in the environment. When an odor finds its particular receptor, this receptor kicks off a series of biochemical events that triggers electrical impulses in olfactory sensory neurons that then go on to send those impulses to the brain. Now, as it happens, each olfactory sensory neuron expresses just one odorant receptor out of, in the mouse, say a possible 1,000 receptors that are encoded in a typical mouse genome. And so we know that mice can smell more than 1,000 odorants. Mice can probably smell about 500,000 odorants. In the audience, we can here probably smell humans between 50 and 100,000 odorants. So there's not a special receptor for every odor in the environment. Instead, we think that odors activate particular combinations of receptors out here in the sensory periphery that go on to trigger activity in particular populations of olfactory receptor neurons that then send that information forward to the brain. So we can use genetic engineering to teach us important lessons about how the peripheral olfactory system organizes information about smells. So this is an experiment where a special protein called the green fluorescent protein, which causes cells to light up green, is expressed in all of the olfactory sensory neurons that express just one out of the 1,000 receptors in the mouse, a receptor called the P2 receptor. And you'll see here, this is the olfactory epithelium of one such mouse. You'll see that these green cells, these sensory neurons, are spread out over the entire surface of the epithelium. But their axons all converge on a single structure called a glomerulus, here with the olfactory bulb. Okay, so all of the olfactory sensory neurons that express a given receptor send axons. They go to the single insular point called a glomerulus in the olfactory bulb. And so there's about 1,000 receptors out here in the periphery, and so there's about 1,000 glomeruli here in the olfactory bulb. Now, a very important point that we've learned from genetics is that the spatial location within the olfactory bulb that any given population of neurons projects to in terms of its glomerulus is the same from animal to animal. It's the same in me and you. And because of that, we can make a kind of map within the olfactory bulb saying where information from any given receptor is going, with P2 going over here and the M71 receptor going over here. And this organizational feature of the olfactory system has an incredibly important consequence. When we smell odors, odors are going to activate specific populations of receptors out here in the periphery that will go on to activate specific populations of olfactory receptor neurons. Those will in turn activate specified populations of glomeruli in the olfactory bulb, as shown here in this functional imaging experiment done by Tom Bose's lab. And because different odors elicit distinguishable and distinct patterns of glomerular activity, with French fries being different from wine, being different from beer, and because the pattern of any given odorant is the same from animal to animal, that's elicited in terms of glomerular activation, with beer looking the same in my olfactory bulb as it does in Steve Lieberle's, the brain can use this these distinguishable patterns of activity that are invariant from animal to animal to identify what odorant the nose is smelling. In other words, within the olfactory bulb, there's a kind of spatial map of odor identity that's composed of these activated glomeruli. This raises the obvious question: how does the brain read out this spatial map of odor identity? Where is the ghost in the machine that looks down on the olfactory bulb, identifies the particular pattern of glomerular activity, and in response generates behaviors, emotions, and memories? In experiments that we've done in my laboratory here at Harvard Medical School, in collaboration with Richard Axel's laboratory at Columbia University, that are actually getting published tomorrow, we've begun to scratch the surface of this problem. And we took the approach of trying to trace the flow of information from single glomeruli in the olfactory bulb forward to the various regions of the olfactory cortex, reasoning that if we could trace the flow of information and understand the patterns of information flow to these higher brain regions, we might be able to discern something about the structure of higher cortical olfactory maps that in turn would tell us a little bit about what these higher cortical areas might be doing in terms of generating odor perceptions and behaviors. And so this is one such experiment. The olfactory bulb is obviously here. On the left, each individual glomerulus is green. You can see all these little green balls. And we've labeled this one glomerulus with this red dye. And we're following the axons that come out of this glomerulus and tracing the flow of information from this glomerulus forward to these multiple cortical regions. And if we do very careful high-resolution analysis of these images, we learn what we believe is a very important principle, which is that there are different olfactory maps in each of these different cortical regions. And we think that the differences between these maps suggest that these different cortical regions actually each are doing different things. So for example, if we look here in the piriform cortex, which is a major part of the olfactory cortex, information from single glomeruli is distributed across the entire piriform cortex. In other words, the insular topographic map of odor identity that I talked to you about in the bulb is completely destroyed in the piriform cortex. We think that implies that the piriform cortex might be involved in processes like odor learning, as in when we learn to be attracted to, say, the scent of a flower when it's coupled with a pleasant experience, like encountering one's wife. In contrast, if we look at projections from the olfactory bulb to these other cortical regions, we see that some of this topography in the olfactory bulb is maintained, that there's hardwired, genetically specified projections from the olfactory bulb to the ventral striatum and this region called the cortical amygdala. And we believe that means that suggests that these areas may be involved in odor-driven innate responses to odors, like the innate attraction a baby has for mother's milk, or the innate avoidance a mouse exhibits when it encounters a cat. So what we think is happening is that, in fact, this insular map of odor identity that's apparent in the olfactory bulb is being radically transformed as information passes to higher cortex. It's being reorganized, not to specify the specific odor identity of the specific identity of any odorant the animal is smelling, but rather to organize information about the behavioral consequence of encountering any given odorant. Our lab and others are now testing this hypothesis. We might be able to understand better how the brain constructs odor maps that let it make sense of the outside olfactory world. And in the very long run, we might be able to use these kinds of experiments to understand how emotion, experience, and expectation impinge on those maps and tweak those maps in order to enable animals to better navigate complex and dynamic environments. And with that, I will stop.
Thank you, Dr. Data. I'd now like to welcome Dr. Margaret Livingstone. Dr. Livingstone is a professor of neurobiology at Harvard Medical School. Most recently, her laboratory has used functional magnetic resonance imaging, MRI, with monkeys to study how the brain processes faces. It's fascinating work. She is well known for her work on visual processing. She has explored the ways in which visual science can inform the world of visual arts. Welcome.
This one? This doesn't seem very nice. Okay. But I don't have my slides.
It's okay, it'll take just 1 second.
Okay. So I'm a neurophysiologist. I try to figure out how we see by recording from neurons in the visual system. But I acknowledge that artists also try to figure out how we see, and sometimes artists discover things about vision that even us neurophysiologists don't catch on sometimes centuries later. So one of the things that artists figured out long before us neuroscientists is that color and lightness, and I'll define lightness, don't worry, luminance, value for those of you in the art field, can be treated separately. They do different things in a work of art. They do different things in our brains. And color, you see, we have 3 cone types. You see color by comparing the activity in 2 cone types. You see light, luminance, by just summing them. It's like black and white photography. And this is important because your visual system is subdivided into 2 major processing streams. One of them we call the ventral stream, we call the what system. It's responsible for object recognition, faces, and color perception. And if you have damage to this part of your brain, you can't recognize objects. Those are called the agnosias. Sometimes you can't recognize something as specific as a face. That's called prosopagnosia. The dorsal stream is called the where system because if you have lesions in that part of your brain, you have trouble navigating through the environment, seeing motion, and spatial organization. Here's some drawings made by people with where system lesions. They were asked to draw a bicycle, a cross, and a clock. You can see that these patients have all the bits right, but the bits aren't in the right place. So what's important for talking about art is that this evolutionarily old where system is completely colorblind. So if you have a red spot on a green background, you could imagine you'd get in your where system or in a black and white photograph, you'd see a gray spot on a gray background. And you could vary the relative brightness of the red in such a way that you couldn't see it in a black and white photograph. That's called equal value, and very weird things happen at equal value. So your ability to see distance or depth is carried by a colorblind part of your visual system. And you see distance the world looks 3-dimensional, right? It is 3-dimensional, I know that, but you see it as 3-dimensional using 2 flat retinal images. So your brain has to go through a lot of fancy computations to figure out distance and depth. It's not like sonar. You don't ping things to see how far away they are. So we use things like relative motion, shading, perspective, occlusion, and the stereopsis, which is the difference in the images in our 2 eyes, in order to compute distance and depth. So I'm going to try to convince you that your ability to see distance and depth is carried by a colorblind part of your visual system. And to do this, I'm going to show you some dots that are moving on a back. Oh, I wish we could turn down the lights. Is that an option anywhere? But I hope this thing looks 3-dimensional to you. It's not, of course. It's flat on the screen, and it looks 3-dimensional whether the red spots are brighter than the background or dimmer than the background. But if I sneak the red dots up so that they're just about the same luminance as the background, it looks flat and squishy, right? Even though thank you. 3-dimensional when it's darker, and then it gets brighter and it gets equal luminant, and it's flat and squishy, and then it gets 3-dimensional again. I hope you saw that. In this image, there's a lot of information about depth from shading, but if we put a red filter over this image and eliminate the blue light and then remove it again, I hope you see more depth from shading in the luminance contrast version. So if you have no luminance contrast, you don't get to see depth very well. Here's another version. What happened here? I went the wrong way. I'm sorry. Another image where I hope that you can see that this thing has a lot of information about depth from perspective. But see if you can even figure out what's going on here. Can you do figure-ground segregation? Do you see any depth at all? And then if we introduce a luminance contrast by putting a red filter in front of this projector, I hope you see more depth from perspective. And the image starts to make some sense. And then lastly, here's an image. This is the Rouen Cathedral. It's a version by Liechtenstein. And it doesn't even look like the Rouen Cathedral until you put a red filter in front of it, and then you can see what he was talking about. So if we put the filters down, thank you, Paula. Appreciate that. So if your ability to see depth from shading is carried by a colorblind part of your visual system, you can't see depth from shading unless the luminance is right, and it doesn't matter what color it is. And so I think some artists played with this. This is André Derain's portrait of Matisse. If you knew Matisse, it probably looked just like him, even though the colors are quite peculiar and don't make any sense. The luminance is appropriate for conveying a sense of 3-dimensionality. And in Matisse's Lady in a Hat, similarly, the planes of her face are bizarre colors, but the shape of her face is there, the reality is there in the luminance alone, and the color is symbolic, just like Picasso said. So I hope I've convinced you that your ability to see depth is carried by a colorblind part of your visual system. Now I want to try to convince you that your ability to see motion is carried by a colorblind part of your visual system. And to do this, I'm going to show you some stripes. We don't have to have the lights down for this one. This is fine. It works just dandy. So these stripes are all moving at the same speed. Wait a minute, I want to do this. So if you look over here, don't cheat, okay? You can cheat by looking at the center, but I'd rather you looked right over here, okay? And if you look over here, and the two stripes seem to be moving at the same speed. I want to get that out of the way. Yeah, there we go. Oops, wrong button. Sorry. Got to get my buttons right. And they look like they're the same speed if the green is brighter than the background or darker than the red. But I hope I can get them to slow down by going right through equal luminance. Now, don't look right at them, and I hope, depending on your equal luminance point, you see them slowing down if you're looking way over here. Don't look over there, okay? So if your ability to see motion is colorblind, your ability to see that something is not moving is colorblind. And I think that a lot of the motion that was achieved by the Impressionist paintings happens when they use equal luminant colors. So Monet's poppies are just about equal value with the background. So your what system can see them, your where system can't. Now, I don't know if the Impressionists did this deliberately or even were aware of the motion they were achieving in their paintings, but I know that Mondrian was because he called this Broadway boogie-woogie. Your what system can see the yellow spots, the yellow checks, but your where system can't, so their position is indeterminate. Advertisers do this all the time. You can't open Wired magazine without seeing something that's got text that's equal value with the background. It jumps around, it's unpleasant to look at, but it catches your attention. So I hope I've convinced you that your ability to see depth and motion are carried by a colorblind part of your visual system. Now I want to try to talk about the fact that color is coarse. You do not have to, that's because the cells that carry information about color have large, low acuity receptive fields, and you don't have to color inside the lines. Here's an illusion based on that. I hope this donut looks yellower than the whole or the background. It's not. This is just a yellow line inside a blue line. Because your color vision, your color perception is low acuity, the color spreads till it hits a border. Now I want to show you some after images to show you your color spreading till it hits a border. What I want you to do is look right at this intersection of these three shapes, and then I'm going to show you a homogeneous gray background, okay? And you'll see an after image. Now look at the intersection and evaluate the after image in this achromatic image. It was better the second time, right? And some of you are saying, oh, it's because you did it twice. Let's do it again. Look at the intersection, evaluate the after image. Look at the intersection, evaluate the after image. Now, this is achromatic, by the way. Look at the intersection and see if you have the color spread to fill a completely different shape. And look at the intersection and see if the color just squirts out the whole. Another thing about acuity that artists figured out, again, long before us neuroscientists, has to do with the Mona Lisa, which is a very famous painting. If you ask the art historians, they say that it's a very lifelike painting because her expression changes. And it changes because her smile is blurry and therefore ambiguous. But I noticed that her smile differed depending on whether I was looking at her eyes or looking at her mouth. So if you look at her eyes and think about how much she's smiling, and then look directly at her mouth, look back and forth between her eyes and her mouth, and see if she doesn't seem to be smiling more when you're looking at her mouth than when you're looking at her eyes. And the reason that I think that this causes the change in expression is because your acuity falls off with your center of gaze. So if you look at this dot, all the letters are equally readable. Your center of gaze is good at seeing little tiny things. Your peripheral vision is good at seeing big blurry things. And actually, your central vision is bad at seeing big blurry things. So if you filter the Mona Lisa in such a way that you see what she would look like to your peripheral vision, or what she would look like to your central vision if you could see the whole thing at once with your central vision. She's smiling more in this than in this. So as you move your eyes around the Mona Lisa, her expression changes, which gives the painting a dynamic quality. And 500 years ago, a dynamic quality in a painting was something very special. You get the same thing in post-impressionist pointillist paintings. Your center of gaze sees the dots, your peripheral vision sees the boats on the river. Chuck Close paintings, your central vision sees the little tiles, your peripheral vision sees the person. Or photo mosaics, your central vision sees baseball cards, your peripheral vision sees Babe Ruth. And lastly, I want to talk about how we see objects with our what system. I want to talk about face perception. We are very good at seeing faces. You're seeing these images in a fraction of a second, and you are not only detecting faces, but you're recognizing individual people. And we can do this whether the images are abstracted, whether they're degraded, whether they're disguised or occluded. And we can do this better than any computer program. We have special regions of our brain dedicated to processing faces. Primates in general do. And we've recorded from face cells, showing them stimuli like these that are sort of faces, but we're varying all the different parameters of faces to ask, what do face cells care about? And we were surprised to discover that face cells, they care about all kinds of these parameters that we looked at, but what we were surprised to discover was that face cells we didn't have face cells that coded for each intermediate, let's say, inner eye distance. Instead, we only had cells that coded for really big inner eye distances or really small inner eye distances. And this was true of all the parameters we tested. If a cell cared about something, it cared about extremes. And extremes are something that artists have figured out, again, long before us neuroscientists. Artists know that we recognize faces by how they differ from the average, because a caricature is much easier to recognize than a veridical line drawing, because face cells code how a face differs from the average face. And even great artists know this. These are images by Picasso, but now you can see who they're of. This is his girlfriend, this is Picasso, Igor Stravinsky, the friend of wife of a friend, his art dealer, his mother-in-law. But they're all caricatures. So vision is information processing. As Bob Data said, our whole brain is about information processing. And you can see how your brain is extracting information from the environment in all kinds of works of art, even art of little children. If you ask a 3-year-old child to draw a person, you will get a face on legs, any 3-year-old child. And this is a drawing that a young child made of his pregnant mother. It's a face with legs inside of a face with legs. What's important about another individual? Their face, right? The kid is telling you that's what he's extracted about these individuals. So artists, either empirically or explicitly, often take advantage of the way our brain extracts information from the environment. Thank you.
Thank you so much, Dr. Livingstone. And now we're going to learn more about sound. And to tell us about it is Dr. Gottlieb Schlaud. He's an associate professor of neurology at Beth Israel Deaconess Hospital and at Harvard Medical School. He is also director of the Music, Neuroimaging, and Stroke Recovery Laboratories at BIDMC and chief of the Division of Cerebrovascular Disorders at BIDMC. Thank you.
So as was already introduced, although I'm covering somewhat hearing, I'm actually choosing the most complex sound that we could hear, which is music. And I'm also combining the hearing system with the motor system because I'm interested in auditory motor interactions. And since I'm a neurologist by training, I'm also interested in how we can use this interaction between the hearing and the motor system and how we can use the most complex form of sounds to help patients recover from dysfunctions. So when we listen to the most complex form of sound that we know, which is music, right. So when we listen to this piece of from Mozart, we not only having an auditory sensation, but it does invoke a multi-sensory. And for those who actually know how to know this piece and know how to play it or know how to play similar pieces, they also have a motor sensation just from a pure auditory input. When we hear music like this and other music, it creates a sense of motion in us, and it certainly triggers our emotional system. And we also know that when we listen to music that we like a lot, that we have emotional connections to, that it activates pleasure and reward systems in the brain. And when we make music over long periods of time, we can actually change the networks in the brain that are related to those forms of music making. And I want to show you one example of this kind of an evolution. Here we have young children that are doing a comparison of two streams of rhythmic sounds. And we either insert an error or not at this second phrase. And while these children process this, we have very strong auditory activation. But the older we get and the more experience we get, even if it is only being passively exposed to sounds or to the regularities or irregularities of sounds, the more activations we can actually see that happens outside of the hearing system. And particularly here, when we get to the adult stage, and we don't have to be adult musicians, even adult non-musicians, even though they're perceiving exactly the same sensations of sound, they seem to activate other areas in the brain that deal with this multi-sensory input that they're actually receiving. And these other areas in the brain also have something to do with making predictions about that sound should actually progress and how the resolution of that sound should be. And it's really these other areas of the brain where there are the biggest differences between musicians and non-musicians, in addition, we have to say, to the auditory cortex, which has been found to be enlarged in individuals that whose auditory input and auditory processing is obviously very important for their profession. These are functional networks in the brain that change with increasing experience or with increasing age, but we also have changes in structural networks. So for example, here in the middle, we see an occasional singer, and we see the structure in the brain that connects the hearing regions to the motor regions that have to do with speaking and singing. And typically there is a slight left-sided advantage of these regions. And obviously it is very important for us when we learn how to speak, when we learn how to sing, that we hear ourselves speaking and singing, and we are able to use that feedback to compare to a template and to make corrections to our output. Now, people that train this all the time, such as singers, actually do have a much more elaborate system on the left side of the brain as well as on the right side of the brain. And probably not surprising to us when we go to individuals that have trouble with singing or that have problems with singing in tune. Is anybody here in the audience who has that? Yes. So there is actually a reason for this. They're missing a particular structure like this one here that is shown in red that actually allows the response to what you're hearing, what you're getting back from your voice, and making so like corrections to your motor output. So like this connection between what you're hearing, a comparison to a template that you may have stored, and then using that kind of a difference between those two inputs and the template to inform your motor system. So for example, a person that really can't sing in tune, if you're going to ask that person to sing Happy Birthday, it would sound like this.
Happy birthday to you, happy birthday to you, happy birthday dear Psyche, happy birthday.
Who thought that was okay? Come on, it should be the same number of people that had their hands raised before. So the typical response would be in people that are so like these out-of-tune singers, or we call them also tone-deaf individuals, that they would say they didn't hear that there was anything wrong with this. It sounded okay to them. And there is a biological reason. Now, if you really think that you have this kind of a disorder, you can actually go to our website and you can take a test. Be really looking for individuals that have this kind of a problem, and particularly if you have family members that have that problem too. So please come and visit us. Now, what I'm interested as well, and which has sort of become an increasing interest over the last years, is whether or not we can, if we have shown, or if we show that music making engages and changes the brain, if it engages the auditory system, if it connects the auditory system more closely with the motor system, then can that be used also in a therapeutic sense? Can we use forms of music making to improve neurological impairments and disorders? And there's actually a number of disorders where this has been tried, and there are varying amounts of data that would support this. I'm going to show you some examples of patients that have suffered a stroke and can't speak anymore but can sing. I'll show you some examples of children with autism that can't speak, but we can use forms of music making to get vocal output from them. And then last but not least, I'll just show you also one form of music making in patients that have Parkinson's disease. So a typical patient with a left-sided stroke that impairs the ability to speak would be this patient here. So this patient has, in addition to the stroke, which you can see here, has actually an interruption of the connection between the hearing and the doing region on the left side of the brain. That is very important for speaking. Fortunately, there is a system on the right side of the brain that could potentially be used for this as well. Now, through development, and because we, for various reasons, primarily use the left side for speaking, this system actually develops quite extraordinary and becomes much larger and becomes much more important. Babies are not born like this. This is a form of adaptation that we all go through by acquiring language and by making these fine nuances that we need to make in order to speak. However, fortunately, there is a system on the right side of the brain that could potentially do this as well. And this is actually a trick that one can use to our advantage to somehow see if we can engage that system on the right side, and we can engage it through forms of singing, and we then can actually use singing to try and help this patient to speak again. When we actually go through this kind of a training process, and when we compare what happens in the brain when we ask individuals to speak, and we're using words that they actually know how to speak and can speak at the outset of our therapy, and then again at the end of our therapy, the major changes that actually happened was that we saw more activation when we compared this one here with this one on the right side of the brain, and in a region of the brain that is the homologue to the left Broca region, to the region of the brain that is directly involved in speech output. And most amazing for us was as well that, remember this rudimentary tract that I showed you on the right side of the brain that is much smaller as a tract on the left side of the brain? We can actually trick the brain as well through very intense and long-term exercise and treatment and practice to change the structure of this connection between the auditory regions and the motor regions of the brain. And this can happen in somebody that is 70 or 75 years old. When we went through this, we were reminded that there might be another disorder where there is a potentially similar problem, and that is in children that are within the autism spectrum but are unable to communicate, unable to speak. And here's one example of this, a child that's almost 5 years old and has not acquired any words but does comprehend. So we couldn't get any words out of him, and his parents couldn't really get any words out of him for the first year of his life. So then we used an intervention that combined sound making, which was actually sound making with his hands, and singing at the same time, because in the brain there's an overlap between hand action and articulatory actions, and the use of singing might actually facilitate the production of words, as we saw in these patients already. So in the first session, we didn't get anything out of him, and we didn't get anything out of him in the third session, and in the fifth session, and the eighth session, but then in the 10th session, something happened, and actually the best one was the 12th session. So we can, through the innovative use of music-making techniques and singing, engaging hands and articulatory actions at the same time, we can make his system get to a point where he can actually make sounds that are meaningful. And just one last example of this, and there has been a lot of interest in using rhythmic auditory stimulation or dance with music in patients with Parkinson's. And I just wanted to show you this little clip that I downloaded from YouTube. A patient that is severely affected by Parkinson, has freezing movements, has dystonic hand posturing, has trouble turning around. And then he walks a little bit out of the camera view, turns on music that he likes, music that has a particular beat that is in agreement with his typical frequency of his gait movements, and then see what happens. All right, so what this actually tells us that the auditory system allows for input to bypass some components of the motor system that are destroyed or that are degenerated in Parkinson's disease to reach regions in the brain that can still help with motor output. It's a very unique component of what we call the rhythmic auditory stimulation in Parkinson's disease. So, music is the most complex form of auditory stimulation. Listening to and making music is not only an auditory experience, but a multisensory motor experience, which has the potential to change brain networks. Music making may have therapeutic potential. There's good data from several experimental trials using music-based interventions that auditory motor impairments can be improved. And last but not least, therapeutic effects seem to last when the therapy changes or rewires the brain. Thank you very much.
Thank you very much. And now we move on to taste. Dr. Stephen Lieberless is an assistant professor in cell biology at Harvard Medical School. He investigates how genes and neural circuits function and affect behavior. Welcome.
Okay, thank you. So smell and taste are our chemical senses, and Bob provided a really nice overview of the olfactory system. Our sense of smell is a cerebral sense. It's a sense of associations. And now I'm going to talk about a more primal sense, an instinctive sense, and that's our sense of taste. So 2-year-olds can relate strongly to our sense of taste, both with positive and negative affect. And this underlies the important role our taste system plays as a gatekeeper to control what enters our body. Taste functions as a surveillance mechanism to analyze the quality of the foods that we eat, both for energy-rich substances, but also to scan for toxins or poisons that we'd prefer not to ingest. And by studying taste, the goal is to get a window into the brain to understand how neural circuits detect chemicals in our environment, and in response, trigger brain regions associated with positive or negative valence. So our tour through the taste system begins with a familiar structure, the taste bud. We have thousands of taste buds in our mouth. Most are concentrated on the tongue, but they're also found in other locations on the roof of our mouth, on the palate. And taste buds contain the primary cells that detect tastes when they enter our mouth, and these are called simply taste cells. We have dedicated taste cells that recognize sweet, that recognize bitter, recognize salty, sour, and savory. And these are our 5 basic tastes. And these tastes not only differ in regard to the different types of cells they activate on the tongue, but they differ in regard to the perceptions they elicit in our brain. And I'm going to walk through each of these taste modalities. So we'll start with the general favorite, which is sweet taste. Sweet is the taste of energy-rich sugars. The sweetest natural chemicals are carbohydrates, like sucrose, fructose, maltose, lactose. And each of these sugars will activate the same cells on your tongue and then the same neural circuits in your brain that give rise to a pleasant sensation. We can also trick our sensory systems with artificial sweeteners that lack caloric value but stimulate our senses. And I want to tell the story of the discovery of saccharin because I think it has a nice moral to it. So saccharin was discovered in 1878 by a synthetic chemist at Johns Hopkins University named Konstantin Fallberg. And Konstantin lived in a time and an era where lab safety regulations weren't as rigorous as they are today. So he was running a chemical reaction and he spilled it all over himself. And rather than clean up, he went home out of frustration and had dinner. So when he had dinner that night, he ate some dinner rolls, and he found the rolls were surprisingly sweet. Now, they had not changed the recipe. Instead, he traced the sweetness to residue from the chemical spill that lingered on his hands and on his lab coats, and he ultimately traced it to a specific chemical that he named saccharin. It's a little scary because many of the artificial sweeteners we use today were actually discovered through similarly serendipitous mechanisms. So the moral of the story is not to eat your lab work. The moral of the story is to appreciate serendipity when it finds you in your experiments. Now that we understand a little bit more about the mechanisms underlying sweet taste, we can imagine new strategies to develop artificial sweeteners with a little more safety and a little more rigor. So I want to take a detour and talk about the discovery of the sweet receptor, because I think this provides a nice example, a powerful example, of how the Human Genome Project and the Mouse Genome Project impacted basic biology and medicine. So the story of the discovery of the sweet receptor comes from some very nice genetic studies where 2 different strains of mice were identified. One could taste saccharin at a low concentration, and another strain of mice that could only taste saccharin at higher concentration. And the ability of mice to taste saccharin at a low or a high concentration was mapped to a single specific location in the genome. When the mouse genome was sequenced, several labs independently asked if there were any interesting genes near this locus. And sure enough, there was a family of taste receptors in the vicinity. Charles Zucker, in beautiful experiments, made genetically altered animals that lacked 2 of these taste receptors, and he found that these animals no longer could detect any sweet chemicals. Also, he showed in beautiful biochemistry experiments that the sweet receptor is a functional complex. It's a 2-subunit complex consisting of 2 of these receptors that could detect every known natural and artificial sweetener. So what defines a chemical as sweet is actually the ability to activate this protein complex. Okay, I'm going to move on to another one of my weaknesses. This is salty taste. The principal ingredient in table salt is a very simple chemical, sodium chloride, and it's the sodium cation that confers the salty taste. Sodium is a key electrolyte in our body. It's found in virtually every cell type at a tightly regulated concentration. And consistent with this, the taste of sodium activates a bimodal taste preference. So at low concentrations, sodium is a pleasant stimulus, but at higher concentrations, sodium is actually quite aversive. So the receptor mechanisms involved in detecting salt are very different from those involved in detecting sweet chemicals. So for the aficionados, sweet chemicals are detected by a family of receptors called G-protein coupled receptors. And salt is mediated, the salt receptor is actually an ion channel that conducts a sodium current into taste cells. Okay, so sour is the other taste of an ion. It's the taste of the proton. And sour obviously has negative connotations in literature. So foods can sour, relationships can sour. A bad event can leave a sour taste in your mouth. So sour taste is the unpleasant taste of acids that provides a warning signal indicating spoiled food and potentially the presence of harmful bacteria. So sour acids will increase the concentration of protons in your mouth, lowering the pH. And all proton-based acids, including citric acids found in lemons and limes, as well as acetic acid found in vinegar, will taste sour. The molecular mechanisms underlying sour taste detection on the tongue are actually pretty controversial, but just like salt taste, it's thought to be mediated by ion channels that conduct a proton current into taste cells. Walking our way through our taste systems, let's talk about bitter. So bitter has the important role of warning us of potential danger. So many toxins and poisons will taste bitter, such as plant-derived alkaloids. Also, we'll add bitter chemicals to many household items, such as detergents and antifreeze to prevent child poisonings, for example. Also, many of my favorite drinks, like coffee that contains caffeine, beer that contains hops, these also have bitter chemicals in them. And first-time drinkers will find these potently aversive, but we can learn to love them, of course. And, and we call this taste conditioning. And taste conditioning actually involves mechanisms that of our more associative senses like olfaction. So flavor is actually the union of olfaction and taste, and olfaction mediates most of the associative components. And even to the most avid coffee drinker, a pill of pure caffeine will still taste potently bitter. Okay, so bitter taste, perhaps surprisingly, involves the same family of receptors, the same superfamily of receptors on your tongue as sweet taste. It involves G protein coupled receptors. But there are some key differences. So bitter cells are different from taste cells. And the sweet receptor is a single protein complex that recognizes all known sweet chemicals. And this makes structural sense because sweet tastes are all carbohydrates with very similar structures. But bitter tastes come in all shapes and sizes. So we've evolved a large family of bitter receptors, each of which can recognize a distinct subset of bitter chemicals. Now, interestingly, a single bitter taste cell on your tongue will express multiple members of this bitter receptor family. So even though two different chemicals which activate different receptors will both activate the same cells, will both activate the same neural circuits in your brain, and ultimately will activate the same categorical sensation. And this is what defines a chemical as bitter. Bitter also, studying the neurobiology of bitter taste is also really interesting because it provides a powerful inroad to understanding the neuronal basis of aversion, and that's something that's poorly understood. And our last basic taste is savory taste, the taste of meat, also known as umami, which is Japanese for delicious. For a while, it was debated whether or not this was a bona fide taste, but the identification of dedicated umami receptors on our tongues really lent credence to this idea, and now it's widely accepted in our field. So umami is the taste of amino acids, which are the building blocks of proteins, and this is sort of the poster child for umami taste. It's glutamate. You might know it as monosodium glutamate, or MSG, which is a common additive in many foods. And interestingly, the receptors for umami are from the same protein family as the receptors for sweet. So it's perhaps not surprising that umami is a pleasant sensation. There are other influences on taste that are poorly characterized. There's the taste of fat, the taste of metal, temperature influences on taste. I bet most of you will agree with me that eating a stick of butter would be pretty unappealing. But if you add fat to many foods, you'll increase the palatability. That's another weakness of mine. Although the mechanisms underlying this are not very well understood. So many basic questions. We don't know whether or not fats activate dedicated taste cells in the tongue, whether or not we detect fats through what's called chemosensation, which is detecting the chemical constituents of fats, fatty acids, triglycerides, or mechanosensation, detecting the texture of fats. Temperature also has a profound influence on taste. We like our meals hot, like a fresh kill. We like some drinks cold. There are chemicals that stimulate our temperature sensing modalities, capsaicin in hot peppers, menthol in cool mint. And these are often additives for food. But we don't yet understand how temperature information is integrated with nutrient sensing information in our mouths. And I want to end, we're running quickly, so I'm just going to make one point that I think the future of taste neurobiology is very exciting, and I think one of the areas which will certainly grow in interest is understanding the architecture of neuronal circuits activated by bitter and sweet taste chemicals, and the relationships between these circuits to understand how reward and aversion are encoded in the brain. So thank you.
Thank you very much. And now for our last sense, we're going to talk about touch. And our final speaker is Dr. Anne Louise Oaklander, who is an associate professor of pathology at Massachusetts General Hospital and an associate professor of neurology at HMS. She directs a laboratory that characterizes neurological disorders that cause chronic pain and itch. Thank you.
Does anybody need a seventh inning stretch? Feel free while they pull up my talk for me. So the senses that we've heard about so far are all the so-called special senses. And they're senses that we've evolved specific organs in order to perceive and process. And what I'll be talking about is sensations that we can perceive widely across our body surface, pretty much anywhere. However, the skin is the body part that is the most specialized for receiving these kind of senses, hence the title of my talk. Somatosensation is what I'll be speaking about. And I've used this picture of the orchestra here, we could perhaps put an X right there, to make the point that what we might perceive as a simple sensation, like, oh, I just touched something cold or something sharp, in fact, as I hope to show you, is the product of a number of different kinds of sensory receptors, all chiming in to give you a perception. So just as when we listen to the music, we may not pick it out, but there could be 4 different violinists and a bassoon and someone hitting a particular percussion only 5 times during the whole piece. And what we perceive is the totality of the experience. You've already seen this drawing of the sensory cortex, how our brain pays attention to incoming sensory signals. And the point is that our brain doesn't listen equally to all areas of the skin. So the parts of the skin that have the greatest impact on our perception of sensation are the face, and particularly the lips, and also the hand, and particularly the index finger, because of course this is what we use to explore our world with. Very interestingly, you'll notice that all of intra-abdominal sensation, the sensations from inside our body, they occupy very little of the brain's attention. And of course, that makes a lot of sense, because who really wants to feel everything you ate for lunch slipping through every inch of your digestive tract? The point also has been made that these senses are profoundly tied in with other very high-level processing systems in the brain, attention. We might notice or hear or listen or feel something at one moment in time when we're paying attention to it, whereas if we're distracted by something else, we could miss something obvious going on right in front of us. Unlike the senses we've heard about so far, there's tremendous diversity in the sensory receptors in the skin. And this cartoon here shows you some of the different major ones, but by no means all of them. This depicts the surface of the skin, or the epidermis. The tissue underneath that is called the dermis. And you can see some of the very different types of sensory receptors for different types of touch. So we'll be speaking in more detail about thermoreceptors that can sense hot or cold stimuli, the Meissner's corpuscles that sense touch, in this case a light touch here of a feather. I'll talk about nociceptors, which is what I work on in my lab at Mass General. These are the free neuronal endings that sense painful stimuli. But even here, you'll notice not all pain is the same thing. So you can have a mechanically painful stimulus, a needle here, or a thermal painful stimulus, such as a burn. The Pacinian corpuscle, I thought this didn't do justice to it, and so I got this larger cartoon showing you that the nerve endings inside Pacinian corpuscles, which are specialized structures to detect vibration, are all wrapped up in these layers of cell processes. When you actually look under the microscope through the skin, they look for all the world like an onion cut in half. And these special layers of membranes enable these neuron endings in the middle to fire if and only if they encounter a vibrating stimulus. We've heard about some of these other modalities. Electrosensation is a highly specialized one that's not that important for humans, but is very important for other kinds of animals. And this slide ties directly into the previous talk because it makes the point that some of the molecules that transduce touch and sensation also affect taste. And these are members of the transient receptor potential superfamily, or the TRPV superfamily as it's known. These are the receptors that transduce temperature sensation. A lot of this work comes from the David Julius lab at UCSF, and they've shown that there's a whole family of related molecules, each of which only opens ion channels in your neurons when the exact preferred temperature of that receptor is attained. But these are multimodal receptors because in addition to transducing temperature, they also transduce some elements of taste. And so the TRPV1 receptor, which is a heat receptor, also is activated by capsaicin, the pungent substance in chili peppers and pepper. And that's the reason why when you bite into something that's been heavily spiced with these peppers, not only will you feel the taste of the pepper, but you'll say, my mouth is on fire, it's burning. It's because it's this same neuronal signal as for burning and for heat. And we heard also that the same is true for cold and menthol, a chemical sensation, which are both mediated via the TRPM8 receptor. I thought it would be useful to maybe explain for a minute how these kinds of sensory signals actually get transformed into the currency of the nervous system, which is electrical activities. And so I brought in what's called a stimulus response curve, which shows you how actual changes impacting on your skin, in this case a change in temperature, changes the electrical activity of different kinds of sensory receptors. So these are free nerve endings that sense heat that are in the skin and in the tongue, as we heard. An interesting point about cold receptors is that it's been found that some of your cold receptors actually are in your blood vessels. And so you've heard the expression, something making your blood run cold. Well, in fact, it does turn out that you can sense cold with your blood vessels. At any rate, the stimulus response curve shows you the temperature of the skin, and so at the beginning of the experiment, it's maintained at a constant, comfortable temperature, and then during the course of the experiment, the skin temperature is briefly lowered and then raised back up to baseline. And a physiologist can make electrical recordings from these different types of neurons and figure out what it is that turns on, that stimulates that particular kind of a neuron. So this cold receptor, for instance, is not firing very much. It's at low frequency at the beginning of the experiment when the skin is at a comfortable temperature, and then when the temperature is dropped, that cold receptor begins to fire action potentials. What you'll see is that it doesn't continue to fire rapidly throughout the entire duration of the period of cold stimulation. In fact, that firing begins to taper off. So that's the reason why when we jump into cold water, a cold swimming pool, for instance, or the brownies, who are the ones who go in the ocean on New Year's Day, It's intensely cold at the very beginning of the cold exposure when the cold neurons are firing maximally, but then if you stick it out and stay in the cold water, it becomes not quite as cold. And at the same time, this other neuron in your skin, that's a warmth receptor, has its activity inhibited when the temperature of the skin is lowered, and it's only in fact when the temperature is restored to warm that that particular kind of sensory receptor begins to fire more rapidly. And the brain is able to decode these changes in patterns of action potentials from different kinds of neurons coming in, and to interpret that into a perception like, ooh, it's really cold in here, or wow, it's so warm I'm sweating, and then to go on and trigger. We spoke about the motor actions. Gee, it's very warm, maybe I'll take my jacket off, or the memories and associations that come with those sensory sensations. My own work, both as a neurologist and as a neuroscientist, centers around the nociffensive sensations. Nociffensive is kind of a funny word. It's really a combination of two words, because these are the neurons that sense noxious stimuli, and these are stimuli that can potentially harm us. So nociffensive neurons are those that defend us against noxious stimuli that can potentially harm us. Because these sensations are so important for survival, you might think that, well, you know, gee, if I couldn't feel any pain, it would be a wonderful thing and how happy my life would be. But the truth of the matter is that when people lose pain sensation through injury or illness, or rare people who are born lacking pain sensation, it actually leads to injury because of the fact that we can't defend ourselves and pull away when we touch something hot or touch something sharp. Just a word about itch. You know, pain, we can all understand why it's useful to have that. But itch is a little bit more difficult to understand, why we would have evolved a separate sensory mechanism for that. And no one knows for sure, but the feeling of most of us who work in this field is that itch evolved at a time when most of us were still very early in human evolution, when most of us were still in Africa, the Middle East, and insects were a major threat. And remember that insects transmit a number of dangerous and in some cases fatal diseases such as malaria. Now, these nociffensive sensations are directly tied into our motor system, and they trigger both reflex actions that occur so quickly we're not aware of them, and then also later purposeful movements. Itch and pain differ because pain triggers a withdrawal reflex, so you pull away from a painful stimulus, whereas when you feel itch, you actually reach out and you scratch or you rub yourself in the area where you feel the itch. And so the thinking is if you had a mosquito containing malaria that landed on your skin, reflexively withdrawing from that would not dislodge the insect. And so that's the reason why we evolved this parallel but somewhat different nociffensive pathway. Chronic pain. So pain is good when you just feel it momentarily, like when you just touch that hot thing and you immediately withdraw from it. The pain will usually last during the period of time when you're recovering from an injury, and it's good there. It's helpful that your broken arm hurts you. Because of that, you don't use the arm, and you actually recover from the injury. The real problem medically comes with chronic pain, which is pain that goes on and on and on, days and weeks and months. And this is a very serious and disabling medical problem. Because of the fact that it's neurons that convey these pain sensations, some of the chronic pain illnesses actually are not caused by problems where the pain is felt, but in the neurons that we've been talking about that carry these sensory messages. One take-home message: shingles is a major cause of chronic neuropathic pain, and it's one that is preventable by vaccines. So everyone over 60 should really be speaking to their physician about the shingles vaccine. Chronic neuropathic pain is hard to detect to know what the cause of the pain is. My laboratory uses tiny little skin biopsies taken from the skin to actually visualize these sensory nerve endings in the skin. And in a series of experiments, we've shown that people who are left with chronic pain, in this case after their shingles, are the ones who have the most severe nerve damage underlying. I'll end by mentioning the reason that we are looking under the skin into the causes of chronic pain is that when we can identify that a patient's pain is neuropathic or caused by neurological problems, instead of just giving them pain medicines, which is fine, but it opens the door to really identifying the actual cause of their pain and treating it. And I'll end on a hopeful note showing you a sad picture of a 15-year-old with complex regional pain syndrome at Mass General a few years ago. And she sent me this very nice picture after her recovery. And so I will end there, and we move on to our next portion. Thank you.